It's a particular and very warm pleasure to me to introduce the first speaker, Rob Semple. It was almost 20 years ago that I received an email from a young Cambridge undergraduate medical student suggesting that he might like to spend a research sabbatical in the United States, and could I perhaps help? And I asked him for his CV, and I received a CV which indicated that this person was about to get the top first in medicine in Cambridge, while also rowing for a rowing blue, which involved 28 hours of rowing practice a week. So I knew I was dealing with a rather extraordinary person. And this has proved true ever since, because Rob's time with Ron Khan in America precipitated his interest in endocrinology, and through working both in Cambridge and London and then Cambridge again, he has developed an extraordinary career as a clinician scientist. Robert has studied both transcriptional regulation of metabolism, reproductive endocrinology, and also now, for the last 10, 12 years, had a big focus, a major focus on disorders of insulin action and its link with growth. And he's been lauded for his successes through prize lectures at the Diabetes UK, at the European Society for Endocrinology, and now with our Starling medal. But that really doesn't do justice to Robert, who's an extraordinarily well-rounded person and human being and physician with great skills as a clinician and wonderful skills as a mentor. I think one of the most sought-after supervisors, mentors, and so respected by his peers and by students that he's now director of the Cambridge MD PhD program. So I'm enormously proud to be able to introduce Robert and celebrate his achievements. And his talk today is going to focus on just one aspect of the brilliant work he does, and that's on the role of activated rather than defective signaling pathways in the causation of human disease. So, Robert.
Thank you, Steve. I think the word I requested was perfunctory regarding my introduction. So you really rather badly breached that. So it is, I'm tremendously honored to be asked to present the inaugural, no less, Starling lecture, but any sense of my own self-importance that I might have after hearing Steve's introduction, I must say was very quickly squashed. Now, being a dutiful chap, the first book I packed for my summer holiday this year was John Henderson's excellent biography of Ernest Starling. And I have to say that as I leafed through it, I progressively became more and more depressed about the paltry nature of all my achievements. Now, Starling was truly a titan of the late Victorian and Edwardian era of physiological experimentation. Were he standing here presenting to you today at my age, he might perhaps tell you about his work on the movement of body fluids and the role of Starling forces therein. He might perhaps describe his discovery of peristalsis together with William Bayliss, but I think most likely he would probably focus on his discovery of secretin, the first bona fide hormone. And by now, some 4 years later, no doubt he'll be using the word hormone, which he's said to have coined over a convivial dinner in Caius College in Cambridge, quite freely. The only mitigation which I can offer for my lack of achievement is that Starling didn't practice clinical medicine after his first year as a house physician at Guy's, and I'm fairly confident he never had to compile an e-portfolio or commission a multi-source feedback. However, even there I'm pushing things somewhat, because he was a man who routinely took up cudgels and fought for bigger issues which he felt very strongly about. And indeed, really, it's only because of those things that he was never rewarded with a knighthood or a Nobel Prize, which both contemporary and subsequent sources believe he richly deserved. And in particular, he was a great admirer of German scientific method and the German approach to physiological experimentation. He became fluent in German during the course of a summer working in Germany as a student. And he used the German example to launch devastating critiques of the Harley Street cabal and the lack of scientific ideal in British medicine. And he was a very strong advocate for things which we now take for granted, such as the creation of medical research units focused around scientists with strong crosstalk with clinical medicine. And he was also a staunch defender of animal experimentation, partly through the bruising nature of the brown dog affair, in which some Swedish suffragettes infiltrated a demonstration and launched a major anti-vivisection protest. And I think had Starling been alive in the age of molecular biology, he very much would have expected today's undergraduates to be able to at least have a conversant knowledge of the signaling pathways whereby the hormones that he discovered exert effects on cells. And in a very crude way here, I've simplified three of the core pathways that emanate from membrane-associated receptors. I'm going to set aside the cytokine group of receptors for now and concentrate rather on insulin IGF signaling, or rather receptor tyrosine kinase signaling, and G-protein coupled receptor signaling. So as a brief précis for any who aren't aficionados, G-protein coupled receptors on binding their ligands leads to the charging of the G-alpha subunit, GTP, which then engages with one of a variety of effector proteins, depending on the type of G protein, to generate second messengers and thereby activate downstream kinases and influence cell behavior. The receptor tyrosine kinases, instead, on being bound by a ligand, autophosphorylate and in turn phosphorylate proximal substrates, which then serve to organize a series of downstream pathways, simplified here as being the RAS-MEK-ERK pathway and the phosphatidylinositol 3-kinase AKT-mTOR pathway. This enzyme, which I'll be talking a lot about, requires recruitment to the membrane, where it acts on membrane-associated PIP2, which is always present, to transiently produce PIP3, which then recruits the downstream enzymes. But before coming back to those, let me first step back in time to the mid-1980s and to what then was the unexplained entity of McCune-Albright syndrome, described by Donovan McCune and Fuller-Albright in 1937. The syndrome, as most of you will know, is one of polyostotic fibrous dysplasia, and then a series of manifestations of activated trophic and effector signaling by endocrine hormones, as well as cafe au lait patches with the characteristic coast of Maine ragged edge. You can see the rather pixelated coast of Maine in the background here, which is in contrast to the coast of California appearance of the smaller patches found in neurofibromatosis. Now, in 1986, before the cause was known, Rudolf Happel, a very distinguished dermatologist, now emeritus professor in Freiburg, suggested that McCune-Albright, as it was never inherited, may well be caused by a mutation which would be lethal if transmitted in the germline and therefore can only be sustained in Mosaic form, where it's admixed with normal tissue. And he elaborated on this idea over subsequent years and extended it to other dermatological disorders. Parenthetically, I also discovered he's a very accomplished and determined writer of German limericks with 2 anthologies to his name so far. And by the time he put forward this hypothesis, progress towards properly understanding G-protein coupled signaling was well in train. So Earl Sutherland, in work on glycogen metabolism in the liver, had discovered CAMP back in the 50s, for which he got a Nobel Prize in '71. Martin Rodbad in the 70s had worked out G-proteins themselves, for which he was similarly rewarded in 1994. And in fact, the same year that Happel came up with his hypothesis, GNAS, encoding the dominant alpha S subunit of G-proteins, was discovered. And it was in fairly short order after that that Henry Bourne at UCSF's lab discovered that in some somatotroph adenomas there was constitutive high-level generation of cyclic AMP, which didn't appear to require hormone. And they duly, a couple of years later, identified the first activating GNAS mutations, really to establish the paradigm which I want to extend to date. And the mutation that they discovered, you can see now in this excerpt from the Sanger Cosmic database, which is simply a collation and a histogram of all the somatic mutations identified in cancer. And you can see the one towering hotspot in the gene, found in around 1,000 tumors to date, is indeed the very mutation they found in their cells, which abolishes or attenuates GTPase activity and leads to persistence of activity of the hormone. And so it was a relatively short step for Alan Spiegel then to adopt this approach and apply it to McCune-Albright syndrome. And in 1991, he reported that the very same types of mutation, indeed, were patchily present in McCune-Albright syndrome. You can see here using denaturing gel gradient electrophoresis, which is akin to using a trowel in a large field to look for a Roman settlement, compared to today's approach with aerial photography and JCBs. So much more onerous than anything we can do now. You can see that compared to the positive control, there was a scattering of mutation signature in each of the tissues they examined. And when they looked at the ovary, in fact, they could see an enrichment of the mutant species in the central part of the ovary, which shows cystic enlargement and stromal hyperthecosis, compared to the rather more normal area at the edge. So that's the paradigm. It's a well-understood signaling pathway. It is activated in the context of cancers, and there is a correlate of cancers, which is the mutations occurring in isolation in mosaic form. So what are receptor tyrosine kinase, PI-3-Kinase, AKT pathway? Well, it followed a fairly similar trajectory. Biochemically, the receptors for EGF, insulin, and PDGF were shown to be ligand-activated protein tyrosine kinases at the start of the 80s. The receptors were duly cloned in the mid-80s. Phosphatidylinositol 3-kinase was described in a viral context about the same time, and then fairly quickly shown to be coupled to activation of the receptors. And then the gene for the dominant catalytic subunit of PI-3-kinase was cloned by the mid-1990s in humans. So far, it's a fairly similar trajectory. And indeed, the next stage was similar too, because around 10 years after that, very common activating mutations were discovered in a wide range of epithelial cancers. And so this is simply the first two publications showing that around 30% of these common tumors have PIK3CA mutations clustered at two hotspots, again echoed now in the more up-to-date Sanger database, which has around 3,000 mutations in the commonest site. And a little bit less common, but still very prevalent in many tumors, were AKT1 mutations. AKT is about 3 steps down the pathway. And similarly, there's one raging hotspot down towards the N-terminal of the gene, which activates the pathway in cells. So that's all very well. And then the next step in the GPCR story would have been to find the corresponding mosaic disease which maps onto those cancer syndromes. But it wasn't exactly clear what that disease was. So we became interested first when we met these three patients. In fact, the boy on the left was a patient described clinically by our chairman's day, some 15 years ago or so now, with a very unusual syndrome of severe infantile hypoglycemia, occurring after about 3 hours of fasting and manifest as soon as mothers try to extend the feeding interval in infancy. You can also see some degree of left hemihypertrophy, and a slighter degree of left hemihypertrophy, mostly affecting the face in this boy, and you'll have to take my word for it, there was also some in the third girl. And you'll also note that each of them has a percutaneous gastrostomy, which was required to keep them alive overnight. And this is a representative example of their biochemistry from their long periods in hospital, where you can see severe spontaneous hypoglycemia, suppressed ketones, normal free fatty acids, low or low normal branched-chain amino acids, and yet undetectable insulin. So in other words, a fingerprint which looks exactly like hyperinsulinism, but with no detectable insulin or any insulin-like substance. Moreover, there is mobilizable glycogen in the liver, and unlike true hyperinsulinism, only a very small trickle of glucose was required to maintain euglycemia. And so, to all intents and purposes, this looked like a syndrome of partial constant insulin excess, but without insulin. And to give you a sense for how challenging this was, this is a graphic taken from the clinical notes of one of the French patients who had a detailed mesenteric venous sampling at the age of 20 months. And the zeros in this column reflect the undetectable immunoactive insulin. And you can see the glucoses at all these different sampling sites are 1.8, 2.2, 2.3, 2.2. It makes me slightly queasy thinking about the nature of that procedure in a 20-month-old, I have to say. So instead of using the trowel and being smart and adopting a candidate gene approach, or at least one in which the candidate gene approach is applied to the actual sequencing, we applied exome sequencing to our proband, first of all, and our candidate gene approach was in silico, applying the human brain to a list of 200 or 300 genes. And what we found was a mutation in AKT2, which I knew immediately was the answer, simply because it's a homologous mutation to that which I showed you in AKT1 in cancer a few moments ago. Not only that, it was present in all three of our probands and in none of their parents. In two of them, it appeared to be heterozygous, and in one was clearly mosaic. So we infer this must have been prezygotic, perhaps in a spermatogonial stem cell, while this must have been shortly postzygotic. And we knew exactly what the mechanism of action of this mutation was from the corresponding AKT1 studies. So they had demonstrated that while the AKT2 enzyme, which actually is the effector enzyme for most of what insulin does in a cell, normally it's exquisitely controlled by the appearance of PIP3, which is tightly coupled to ligand activation and only appears when insulin binds to its receptor. But in effect, this was somewhat relaxed, and so the new mutant AKT2 had some ability to interact with PIP2, which is present all the time. So in other words, the coupling to upstream ligand stimulation was slightly reduced, or it was slightly attenuated. And the biochemical consequences really were rather mild. So this is some blots from primary patient cells showing that compared to cells on which a large amount of insulin is put, there is very, very little basal activation, but there is basal activation nevertheless to a very small degree. You can see that rendered alternatively over here using phospholysers, where the critical difference causing this disastrous syndrome in the children is the difference between this and this. This is one of the phospho-AKT sites, and this and this. There's also, incidentally, a very reliable and persistent reduction in the peak phosphorylation, which is achieved after insulin. So biochemically, this is a very small change, but clinically, it's catastrophic. And in a second in vitro experiment, we showed that the presence of this mutation when overexpressed in fat cells was enough to abolish the ability of the transcription factor FOXO1 to enter the nucleus in the absence of insulin, where it turns on gluconeogenic gene expression. You can see it's tonically excluded in the presence of this mutation. This is a very rare entity, and only 2 subsequent patients have appeared, one reported by Khalid Hussein from a patient in Austria, and a further patient described this year from Turkey, where they made a little bit more of the facial dysmorphism, which I'm inclined to think is a gestalt, because our patients too have some of this proptosis and possibly some ptosis as well. So I think as we see more patients, we will gather more understanding. But their metabolic profiles were identical. And while we were working on our AKT2 patient, Les Biesecker, the year before, published in the New England Journal precisely the homologous mutation in AKT1. So this is the cancer mutation in Proteus syndrome, a horrendously disfiguring syndrome believed to have affected Joseph Merrick, although not yet proven, I understand, as his bones have been boiled in bleach several times over the years. AKT3 is the isoform which is mostly found in the brain, and again, the same homologous mutation present in Mosaic form leads to hemibrain overgrowth, and you can see this leads to epilepsy, which is often sufficiently intractable to require hemispherectomy, as you can see here. So our first obligation is simply to understand the natural history of this condition more. So we continue to track our patients. And this is unpublished data just showing a recent evaluation of the 2 boys who are both born within a week of each other. They're both 17 years old. They both have 37% body fat, in other words, have become quite obese. And you can see that at the age of 17, one of them persistently becomes hypoglycemic overnight when left to his own devices, requiring rescue with Lucozade at this point. And at the time of hypoglycemia, he has undetectable insulin and low free fatty acids, which moreover show none of the normal nocturnal nighttime excursion. While the second boy appears almost to have auto-corrected, and while his insulin levels remain very low overnight, he is able to go through the night with a normal glucose and with evidence of lipolysis occurring as expected. Their glucose tolerance tests, despite that cellular evidence of mild blunting of phosphorylation, are relatively normal. So it's interesting to speculate why one patient is auto-corrected, but I have nothing more but speculation to offer now. So our eyes were well and truly into this business of somatic activation of the signaling pathway when we met this lady who was sent down to us with the suggestion raised that she might have lipodystrophy affecting her top half, and this may be her dominant problem in the context of many years of desperation in trying to find an etiology for her disorder. And you can see that that disorder is one of enormously overgrown legs with a persistently gaunt appearance of the upper body. And critically, when we examined her abdomen, we could see that this was asymmetric, so this wasn't a plain craniocaudal gradient. Looking at her abdomen, you can see there's lots of fat on one side of the abdomen and none on the other on the CT scan, and you can get a sense of a midline difference there. So it was very difficult to conceive of this as anything other than a mosaic disorder. So we duly exome sequenced DNA from her arm and from her leg, and we asked the question, were there activating mutations in the leg which might account for this overgrowth? And there were. We had, first of all, to get rid of some false positives and false negatives arising either from the sequencing or from mutations which had occurred in older cells in culture. But we ultimately boiled things down to 2 mutations which were truly present in leg but not arm. And one of those was the canonical activating P110 alpha mutation, which sits in the catalytic subunit of PI-3-Kinase, which I showed you on that histogram a few slides ago. We had, I think, been a little bit lucky that we picked that up so easily, because in the cells it appeared heterozygous, where it had probably been selected for ex vivo. While when we went back to the tissues, we had lots of little blips, which are the sign of mosaicism, and which have caused me no end of worry since then, given the number of blips on sequencing chromatograms I've ignored over the years. And as might be expected, we found that in cells from this lady and similar patients, the levels of PIP3 at baseline with no hormone present were equivalent to those of wild-type cells which were stimulated with hormone. And furthermore, when the mutant cells were stimulated, they developed an amplified production of PIP3. This was seen also in activation of downstream pathways. This is an ELISA to look at phosphorylation of AKT, and in Western blots where the wild-type cells have no phosphorylation at base time, and you can see a messy variable phosphorylation in the patient cells. So very quickly, we worked with Les Biesecker and assembled a cohort of patients who had broadly similar problems in the sense that they were what we called fibroadipose hyperplasia, but affecting different parts of their bodies. And so you get a bit of a sense of the spectrum which we initially discovered here. And you can see also that while some patients are very mildly affected with just a single digit being enlarged, and while some are rather horribly affected up here, the actual types of mutations are the same. And so in any Mendelian disease, although we know that disease genotype is probably the dominant determinant of phenotype, we quickly formed the view that actually in these disorders, it's the time and place of the founder mutation that was the key determinant. However, this is a very valuable paradigm in this field and more generally in the field of rare disease. And it's an Indian parable rendered here by John Godfrey Sachs, who was a Vermont poet. And he tells the story of a group of blind men who go to investigate an element by palpation, if you will. And they come back each having held a different part of the element and argue very volubly about what they've just discovered. The point being that although each was partly in the right, all were in the wrong. And if ever there was a field where there's a risk of this happening, it's this field here, given such diverse clinical presentations. And all this was discovered only 3 years ago now, and really the whole area has exploded and thrown into the melting pot a wide range of patchy overgrowth disorders. And that's a process which is still going on, and we should be fairly agnostic about classification until the process plays its course. At the very same time that we published our paper, others published papers describing the same types of mutations in patients with hemibrain overgrowth. And in short order since then, similar mutations have been described in a wide variety of syndromes much beloved to geneticists in particular. You see some fairly well-known entities in there, such as Klippel-Trenaunay syndrome, as well as a lot of rarer syndromes. And so we've been mucking in and doing our bit by finding as many patients as possible and working out the disease spectrum. And this is already slightly out of date because we now have about 230 patients who've come to us in the course of the last 3 years. But to this point, our diagnostic algorithm involved, first of all, skimming off the patients with the hotspot mutations, those ragingly high points on the histogram, which we do with the restriction fragment assay. And then applied next-generation sequencing to a focused panel of related genes, which again picks out a lot more, and in fact will now pick out far more than shown on this algorithm as we've improved our panel. And the mutations we're finding are not just in PIK3CA itself, but in other pathway genes. In some patients, we're left with no causative variant, and the question there is that simply because this is a patchy disorder and we haven't sampled affected tissue, or is it because it's a more novel gene which isn't in our pathway? And we very much bear in mind our signaling studies in those cells and have a lot of confidence if we do see pathway activation there. So I would eschew any overzealous use of any of the clinical descriptors which you've seen before, but I think we can say that the nature of these disorders is that they each feature an overlapping combination of different pathological features, which include very prominent vascular malformations seen here on the skin, but also large vessel malformations internally. Occasionally, muscular overgrowth. This is a Danish girl with enormous hypertrophy of her trapezius muscles. The brain, as you've seen, can be affected. There's enormous overgrowth of peripheral nerves sometimes as well. This is a median nerve from a patient in Texas, and we've seen this in several other patients. And it raises the interesting question of why we manage to get such a lot of limb overgrowth, often in the face of very small mutation burdens. And so I'm not sure if there's a paracrine effect here, or if there might be a neurotrophic element to some of the limb growth, a sort of opposite of what's seen in thalidomide poisoning. You can see that adipose tissue is very commonly severely affected, probably simply because it is one of the most plastic tissues and can expand most easily. And there's often a nasty destructive arthropathy as well. So you may be asking, what's in there for endocrinologists? Well, these patients do sometimes come to endocrinologists, especially the patients with the milder, more diffuse mutations who present with primary growth disorders to pediatricians. Sometimes they also do present with hypoglycemia, and it's hypoglycemia of exactly the same type that I described before in the context of AKT2 mutations. Interestingly, we've never seen that in any of those grossly affected patients with large limbs. We only ever see it in the patients with mild diffuse overgrowth and what we believe to be milder mutations. And I think that's because only in those patients can the liver tolerate a large mutation burden. And you can see in this girl from Germany who has the phenotypic features of what's called macrocephaly with capillary malformation syndrome, also has a profile of hypoglycemia, apparent on the first day of life with undetectable incident and suppressed ketones. And she's following a similar course to those other patients whom I described with the AKT2 mutation. So this is all work in progress, but we progressively are accumulating more and more mutations, not just in PIK3CA, but also in other parts of the pathway. Mutations are almost never seen in the germline or in lymphocyte DNA, and certainly the strongly activating mutations, which I take to be these hotspot mutations, I've never ever seen in the blood, so I think Happel was essentially right. Some of the rarer mutations, which we think are more mildly activating, can sometimes be tolerated as mosaics in the germline. And there is just a hint of a genotype-phenotype influence in that the strong mutations give rise to those very strikingly asymmetric regional overgrowth syndromes, whereas the mild ones, as I've said, often give rise to a more lumpy, mildly asymmetric overgrowth, sometimes with hypoglycemia. And interestingly, it seems that the excess cancer risk is barely detectable in the several hundred patients that we know about so far. Now, one of the most dispiriting bits of doing rare disease research and looking for new genes is it's very rare that finding a new gene actually gives rise to a new treatment. And this is one of those rare cases where we may have a chance. And that's because this pathway has been known to be activated in cancer for the last 10 years. And as well as the mTOR inhibitors, sirolimus and similar agents, there are a range of different agents in cancer advanced trials targeting some or combinations of different elements of the pathway. So we think we have an exciting opportunity to try to influence natural history here. The experiments of putting these inhibitors on cells in some ways is slightly trivial. It's obvious that they're going to turn off the pathway. But I think what's encouraging is that we find that everolimus, which is a prodrug of sirolimus, can turn off the excess signaling at tiny, tiny doses. So we think we may be able to get away, potentially, with a pharmacodynamic effect at very, very low doses. And we see this beneficial effect across a wide range of different cell types with different mutations. And you can see it maps onto even greater effects when we start to look at some downstream elements of the pathway. But at present, we simply don't know if it works. Because it's such an obvious thing to do, increasing numbers of people around the world are publishing small case series and putting together small trials. We have put together a few patients who have received sirolimus on the grounds of clinical need, including this lady, the first lady we met, who chose to take sirolimus some 4 years ago now. It's very uncontrolled, unblinded, but you can see that her body fat mass, as estimated by DEXA scanning in her legs, was around 60 kilograms at the time we started this in 2012, and has fallen to a fairly dramatic degree in terms of that DEXA estimation of body fat. And that correlates with the fact she's now able to wear different prostheses, which she grew out of a long time ago. So we're somewhat encouraged, and you can also see this epidermal nevus at the base of her back, at the top of her nasal cleft, dramatically improved over the course of treatment as well. We also would very much like to try some sirolimus in the patients with hypoglycemia, but all the parents we've come across so far have been rather sensible, and they said, look, we can do this with modified starch and with percutaneous feeding, and nobody has felt bold enough to try sirolimus yet. It's also more difficult to produce the preclinical proof of efficacy. This is the patient with hypoglycemia and the PIK3CA mutation that I showed you before, and you can see the level of basal activation is very slight, but the same argument applies that it's likely that that small level is enough to be clinically very manifest. Nevertheless, it does turn off with sirolimus, and we have considerable interest as time goes by in looking to see whether hypoglycemia can also be improved. So you might think this is almost a done deal. Why not just use sirolimus and do this properly with randomized controlled trials? And no doubt there will be some trajectory towards that. We have just one note of caution, however, and that is that when we put sirolimus on patient cells, we see that the PIP3 levels are boosted by feedback processes which are known about but haven't been really well worked up, and the mechanism for which isn't understood. So you can see that everolimus applied to mutant cells gives a bigger rise than patient cells, and this is really quite a spectacular rise by the time you combine that with a ligand. And it corresponds with activation of downstream signaling. It is squelched to some extent by farnesyl transferase inhibitors, so RAS may be playing a role, although it's a very dirty sort of inhibitor. And it doesn't seem to be driven by the P110 alpha itself. Now, I just don't know if that's an irrelevant cellular artifact or not, but there are processes in the cell which are PIP3 dependent, but not AKT dependent, such as activation of the small G protein RAC. And you can see that in this summary slide showing cancer mutations, as well as the PI-3-Kinase mutations. I won't go into the statistical details of what this shows, but suffice to say that mutations appearing, or genes appearing up towards the top right, are the most strongly associated with cancer. As well as this cluster of PI-3-Kinase pathway mutations occurring up here, RAC1 is also there. And of course, in these disorders, we may potentially want to treat children lifelong with small doses of inhibitor. So I think it's very worthwhile working up the alternative approach of specifically targeted PI-3-Kinase inhibitors, and we're in the process of forming collaborations with interested pharmaceutical companies to do that at the moment. You can see for at least one of the compounds which we've evaluated, they too, unsurprisingly, are very efficacious in turning off signaling in the cells, and actually in restoring some dynamic range to signaling, in other words, suppressing the basal activation, but increasing the peak activation because of diminution of negative feedback pathways. So I'm fairly close to being finished. There's a huge number of scientific questions we have about these disorders. We want to know a lot more about the timing of the mutational event, the cell of origin, and discover which lineages can bear these mutations and in which lineages it's selected against, such as the hematopoietic lineage, for example. The Happel hypothesis does broadly hold. There is some genotype-phenotype correlation. We do see odd mismatches between mutation burdens and degree of overgrowth. So I think these questions of neurotrophism or paracrine effects of activated cells are important. Most important from the patient's point of view, we want to understand how much of what we see is irreversible developmental abnormality and how much is persistent reversible dysfunction. We think the cancer risk is low, but we critically want to know if we can make anything regress with the inhibitors, which I've talked about. And more generally, I think the whole issue of mosaicism is now very much to the fore, and people are actively looking around to find out which other disease states mosaicism might play a role in. So I'm actually not going to go through the summary, which I think I've dealt with in some detail, and I'll deal instead with my acknowledgements. Now, let me be quite clear, I'm not making any socio-economic points here. I'm simply very conscious that I'm here today only because of the mentorship I've had from our chairman, Steve, who is not only scientifically perspicacious, but he's very generous, and has had a very unusual style of mentorship where he's allowed more than one of us to continue to thrive and grow, and frankly usurp a little bit of his academic space in Cambridge, and that's testimony to why we all get on so well together and enjoy our time there so much. And of course, all of this is flavoured heavily with his bon mot and his, I have to tell you at times, pithy profanities. And the other person I need to thank in particular for this work is Vicky Parker, who is a Wellcome Trust training fellow and continues to work very hard in this area. She had the good luck, first of all, to be in the lab just as we found the first mutation, but I can't emphasize enough how she has taken up the baton and run hard with it and shown formidable organization in setting up networks of patient recruitment and diagnostic algorithms and forming liaisons with pharmaceutical companies. And it simply wouldn't have been possible without all her work, and I'm sorry she hasn't had the chance to present it herself today. And we can't trace the lineage all the way back to God, but we can perhaps trace it at least back to Ernest Starling, if only because of his strong views in what we now all take to be granted, the importance of integrated units of scientists working together with clinicians, sometimes the same people, sometimes not, in a highly fertile environment. And I hope that he would have approved of the environment that so many of us work in these days. And very finally, it would be outrageous not to mention the very large number of other people who are involved in this. I've highlighted some of the key people in yellow here. Ben Chalice is here at the conference today. He's played an important role in the hypoglycemia work, together with Nuno Rocha, Rina Minic, and Sarah Leiter. Our collaborations with the Sanger and the Babraham Institute were very important for our early genetic discovery and workup. But increasingly, the landscape is now dominated by the growing network of excellent partners and collaborators with whom we're trying to extend our clinical trial efforts and our natural history efforts as well. Thank you.